Hello, my dear students. Welcome to my today's online class on gene therapy. All of you know that we have somatic cells or body cells and gametic cells or sex cells. And in these cells, you find the nuclei. Inside the nucleus, you find the genetic material in the form of DNA. So this is about DNA or the gene concept. A gene is basically a segment of DNA that codes for a functional protein. Sometimes this gene can be defective in organisms when it doesn't produce the functional protein. As a result, the individual with this bad gene will suffer from disease. And this bad gene can be replaced by a functional gene, and that is known as gene therapy. Okay, this is the topic for our class today. And I am Dr. Meera BK, Associate Professor and Head, Department of Zoology, Maharani Science College for Women, Palace Road, Bengaluru 1. You can see my cell phone number and mail ID on the slide. And after the class, to clarify any doubts, you can contact me. So this diagram in a nutshell tells you what is gene therapy. Here you have a cell with non-functioning gene so that the functional protein is not produced. And the bad gene can be corrected by replacing it with a functioning gene or good gene, which will be able to produce the functional protein. And once this gene therapy is done, the cell will start functioning normally. This is what exactly the gene therapy is. Now, so gene therapy is the process of inserting genes into the cells to treat the diseases. The newly introduced genes will encode functional proteins and therefore correct the deficiencies that occur in the genetic disease. Thus, gene therapy involves genetic manipulation in animals or humans to correct a disease, and ultimately the organism is kept in good health. So same diagram I have given here. Then there are two approaches for the study of gene therapy. One is somatic cell gene therapy. The non-reproductive cells of an organism, body cells, are called somatic cells. For example, bone marrow cells, blood cells, skin cells, intestinal cells, etc. They are all somatic cells. This gene therapy, that is somatic cell gene therapy, is directed to correct the genetic defects that are found in somatic cells or the body cells. The therapy involves the insertion of a fully functional and expressible gene, in the sense that produces a functional protein. Okay. So insertion of a fully functional and expressible gene into a target somatic cell to correct a genetic disease permanently. The genetic alterations in somatic cells, please note, they are not carried to the next generations. Okay. Whereas if gene therapy is done at germinal cell lines, it is heritable, it is carried on to the next generations, which is not so in case of somatic cell gene therapy. Then this is preferred extensively studied with an ultimate objective of correcting human diseases. All right. So now here you can see a schematic diagram. Here we have a ADA gene. Okay. If this gene is defective, I'll come to it later. If this gene is defective, the individual will suffer from a disease known as SCID disease, severe combined immunodeficiency syndrome. Okay. Now, this good gene, functional ADA gene, can always be cloned into a virus. Okay. So that you will get a cloned ADA gene which is incorporated into virus, and then this viral vector can be treated with disabled T cells which are disabled with ADA gene isolated from SCID patient. Okay, the patients will have their T cells. T cells are a type of immune cells, and these defective T cells will not have or will have disabled ADA gene in them. So these T cells with disabled ADA gene are isolated from SCID patient, and you can give them viral vector which is cloned with normal ADA gene, and so that when retrovirus infects these T cells, it transfers the normal ADA gene into these cells. So these cells which have incorporated normal ADA gene into them by means of retroviral infection, they can be grown in a culture, and these cells can be given to a human being. So genetically altered cells are reimplanted in the human beings to produce ADA. As a result, the person will be cured from SCID. He will be normal with his normal immune system. This is what is exactly done in somatic cell gene therapy. So this is the best example. SCID treatment, gene therapy for SCID is the best example for somatic cell gene therapy. Okay. Then the second approach is germ cell line gene therapy. Here, the reproductive cells of an organism, like gametic cells, constitute the germ cell line. Gene therapy involving the introduction of DNA into these germ cells, okay, is called germ cell gene therapy. And please note, it is passed on to the successive generations, which doesn't happen in somatic cell gene therapy. But for safety, ethical and technical reasons, this therapy is not being attempted right now. Okay. Now, this slide will tell you what exactly is done in germline gene therapy. See, here you can see a couple. They are the parents with genetic disease. They have bad gene in their constitution. So the male produces the sperm and the female produces the ovum, and the sperm fertilizes the ovum to produce a zygote with mutant gene, with bad gene or with dysfunctional gene. And this zygote, as usual, undergoes cleavage, blastula, etc. to form a cell culture. And please note, these cells have mutant genes, okay? Bad gene in the sense, disabled genes. And the baby which is the product of this blastocyst, is going to develop the genetic disease. This is what the normal process. But what you can do is, at the blastocyst stage, that is embryo with the genetic disease, at this stage, you can take the cells with bad genes and put them to culture. You can subject them to cell culture, you can treat them. And on the other hand, you have a viral vector containing the therapeutic gene. Okay, viral vector which has incorporated a functional gene in it, and even that is added to the cell culture. And then you will get a genetically modified cell from the culture, where the bad gene has been replaced by functional gene. And now what you have to do is, you have to take the ovum with the nucleus removed. You have to take the ovum without the nucleus, that we call enucleated egg cell. You can club the enucleated egg cell and genetically modified cell from the culture, and you will get a genetically improved zygote. You will get genetically improved zygote, and the embryo developing out of this zygote, will be normal, and it is going to, and the baby coming out of this improved embryo, genetically improved embryo, is going to be a healthy baby without any genetic disease. This is what exactly is done in germline gene therapy, and please note, this is passed on from generation to generations. Okay, then. So this PPT will brief the differences between somatic cell gene therapy and germline gene therapy.